Welcome, ladies and gentlemen, to the 2019 Oregon Wine Symposium session on the microbiome. For the next hour, we will explore what is known about the wine grape microbiome and its impacts on the wines. We have three speakers today. Dr. Jeremy Wise of Linfield College. He has a long interest in the microbiome of sponges, and for the past 5 years, he's extended that interest to the vineyard microbiome and how it's reflected in the winery microbiome. His work is funded by the OWB, and he will describe the latest results from those studies. Dr. Krista McGuire of the University of Oregon will discuss the work of her lab on the vineyard microbial ecosystem. She has a long interest in the interactions between plants and microbes and how anthropogenic interactions, such as those in agriculture, alter biodiversity. And Greg Jones, holder of an endowed chair at Linfield College and a scientist for all seasons in our industry, pun intended, will talk about the place microbiome in our concept, has in our concept of terroir. Well, what is the microbiome? Broadly defined, it's the entire small world of bacteria, fungi, and archaea that grows in the vineyard. Technically, there may even be some mosses and algae in there as well. These microbial components may be located on leaves, fruit, flowers, wood, and of course in the soil, which is likely to function as the major reservoir. But there may be other reservoirs as well, such as in neighboring vegetation, upwind orchards, and the like. How does one detect the microbiome? Well, this is really a critical issue. The classical approach would be through culturing of microorganisms on bacterial or fungal specific medium in a Petri plate. The limitations of this method would be whether a specific organism would grow on that medium, whether it would grow by itself, or whether it would grow only in its original habitat, say on grape skins. The point is that by this method, one can only detect what can be cultured. Many microorganisms only like to grow in commensal or interdependent environments. Many or possibly even most organisms in the wild are like this, and we might not be able to detect them by classical methods. Modern methods are based on detection of nucleic acid signatures, either by looking directly at the nucleic acids extracted from a sample or by amplification of small amounts of nucleic acid in the test tube. Very large databases have been and are being built to enable us to identify those nucleic acid signatures and thus facilitate this approach. Well, this is all lovely, and why should we care about the microbiome? Well, the microbiome was a significant topic of discussion at the International Terroir Congress held at Linfield a few years ago, and there is increasing evidence that for a microbial component of terroir in our vineyards, the regional flavors and aromas in wine may be due in part to regionally structured microbial diversity. Thus, the vineyard microbiome could be expected to drive the relationship between the managed and unmanaged diversity in the winery and in the ferment. Even though microbial diversity might narrow considerably during the course of fermentation, the qualitative signatures of members of that initial community, that initial microbial community, might well contribute to the characteristic wine attributes. Furthermore, understanding microbial diversity and its trajectory might help make sense of the effects of cultural practices such as sustainable farming or biodynamic farming, etc., on wine characteristics. Now, let us spend the rest of this hour exploring some of these ideas in depth, and let us call to the podium Dr. Jeremy Wise to begin our exploration. Jeremy?
Good morning, everyone. Thank you all for being here. Can I get my PowerPoint up, please? Thank you. Yay. All right. I wanted to start out this morning with my acknowledgments so that I make sure I get them done. Like any academic, most of my work is not done by me, but done by students. So this work is primarily done by Shay Geisher, who's a recent Linfield graduate, and Lexi Morris, who's here today. And lots of students have helped out with collections. And I also definitely want to thank all the vineyard owners and managers who let us come onto their property and take some grapes. And as David said, this is an Oregon Wine Board funded project, so thank you to all of you for letting me do this project. So, as David just said, right, terroir is formed at least in part by microbes, right? We use the term microbial terroir, this sort of vaguely undetermined, as yet undetermined thing. And one of my big goals is to try and figure out what that means. Like, what does it mean to have microbial terroir? So all the data all the research that has been done on this thus far has shown that every vineyard has a unique microbiome, has a unique microbial terroir. But what I'd really like to start to tease apart is what causes that microbiome to be different. A couple early studies have been done. There's really not a lot that's been done on this. This is a figure from one of the first studies that was done. This is from 2014. And you can see the title there, right? Microbial biogeography is conditioned by cultivar, vintage, and climate. But one of the biggest patterns that they saw was geographical, right? So Central Coast vineyards had different microbiomes than Napa and different than Sonoma. I should say at this point, no one except for Dr. McGuire and I have done any of this in Oregon, so you're looking at some of the only data that exists for your vineyards. So we know that geography drives things, but what I was really especially interested in is, are there things that you all do in your vineyards that influence the microbial terroir, that influence the microbiome? So specifically, are there farming practices that have an effect on what microbes are present on grapes, and therefore what microbes actually go into a vineyard? So what do? We went out to several vineyards, and I'll get to that on the next slide. And we go out and we collect grapes. So we just go down rows and we sample entire clusters. We keep them all sterile, bag them, bring them back to Linfield, and my students process them, right, mash the berries up, extract DNA. We get it sequenced at OSU, and then we bring that back and analyze the samples. So this is a picture of Lexi doing some sampling, and I just have two other pictures too. And I want to say this is definitely a group effort. So the left picture there, the little girl there is my daughter, because when you need people to help you sample, you take whoever you can get. My daughter was really excited, so I brought her out, and she helped. And it's also a perk of doing this job, is I get to go wander all of your vineyards and see all the pretty views and taste your grapes and things like that. Okay, so we went out. I've been doing this for several years. I want to share 2017 and 2018 vintage data with you. So we went out in 2017 and we sampled in two AVAs, the McMinnville AVA and the Aola Amity Hills AVA. And in each of those AVAs, we sampled two vineyards, one that was biodynamic, so certified biodynamic, and one that was not. So in this case, they're both sustainable. I'm not naming vineyards. You can probably make guesses on them. I will say that CB is Crawford Beck, so it's David and Jean Beck's vineyard. So those 4 vineyards in 2017. We go out, we take all the grapes, we process them, and what do we find? So in 2017, if we just look at straight diversity, so sort of how many bacteria are present there, we found a significant difference in diversity between biodynamic and the non-biodynamic vineyards. So as you can see here, the biodynamics had significantly higher diversity. Whether that's good or bad, we don't know, but it's different. And if we look at between the AVAs, we see that there was no significant difference in diversity. So this was a bit surprising to me, right? We know that there's differences between AVAs, geographic differences, but we did not see that. Granted, these are essentially neighboring AVAs, so there's not a lot of geographic differences between them, but we saw no differences. All right, but that's just a rough measurement. What we do then is we make these figures that are called NMDS plots, and they're a little confusing, so I'm going to take a minute and sort of talk you through it. So we take all of this data, right? So I take a sample, I get back some amount of information on the community of bacteria, in this case, that are present there. So it's a big, huge community. I have abundances of like several hundred species of bacteria that are present in every sample. What this figure is it takes all of that data and simplifies the community down to essentially a point on a grid. So every vertex there, every corner of each of those triangles represents a two-dimensional simplification of the community. Then I can connect points together that overlap. So in this case, right, they're labeled McMinnville conventional, Aeola amity conventional, and then biodynamic from McMinnville and from Aeola amity. And generally speaking, as we look at these triangles that overlap do not have different communities. Triangles that don't overlap do have different communities. So 2017 data, we can see that the two conventional non-biodynamic vineyards overlap, the two biodynamic vineyards overlap. So across two different AVAs, the driving factor here in sort of shaping microbiome, microbial terroir, is the farming practice more than the geography. All right, so this is good, but that's not a lot of data points, so we wanted to expand this. So in 2018, I'm now a WB-funded researcher. We went out and we looked at 5 AVAs. So Ribbon Ridge, Yamhill Carlton, McMinnville, Dundee Hills, and Aeola Amity Hills. And we sampled, from each of those AVAs, we picked 2 vineyards, one that was biodynamic and one that was not biodynamic. I'm referring to them as conventional, but that's really an oversimplification, right? Again, a lot of these are live certified or otherwise sustainable. So really I'm distinguishing based on things that are biodynamic, so only non-synthetic inputs versus non-biodynamic have some synthetic inputs. So that's my list of 10 vineyards, right, all nicely paired. As we got back our data, which was over the weekend, the biodynamic from Aeola amity, we did not get good results on. So I ended up with 9 vineyards from 2018 that we can look at. And we can do the same kind of analyses. So first, diversity. We just look at sort of broad patterns here. At our larger data set, now we're not seeing differences in diversity among different farming practices. So not showing that here, but the biodynamic versus conventional diversity is not all different. If we look at AVA differences, so geographic differences, now we're seeing a little bit of difference, but mostly it's just Aeola amity that is significantly higher diversity than everything else. And because that's the AVA that we lost a sample from, that actually is just Crawford Beck. So I don't have an explanation for that, but apparently Crawford Beck has the most diverse of all the ones that we sampled. All right, but we can look at the same kind of figure now. So again, another NMDS plot. This is a much bigger polygon, a much bigger shape there because I have a lot more samples. And they do overlap some, as you can see, but the statistical tests revealed they are significantly different, which means that, again, we're seeing big, significant differences in the microbiome, in the microbial terroir of these grapes, driven by farming practices, by presence or absence of synthetic inputs. If I take this data and plot it by AVA, it looks like this. It's kind of messy. There's a lot of overlap there. Again, the only thing that stands out, right, we do have significant differences, but the only thing that stands out is Aeola amity. So again, it's just that Crawford Beck vineyard that's different. Otherwise, we're not really seeing differences based on geography. So now, even on a wider scale, right, and this is still just all essentially North Willamette Valley vineyards, but a wider sampling of those vineyards, we're not seeing differences based on geography. We're only seeing differences based on farming practices. Okay, so I wanted to dig a little bit deeper and see, can we figure out what's sort of driving these differences? So I did an analysis that's called LEFSI that allows me to identify what are the biomarkers? What are the species strains of bacteria that are driving these differences. And what I found is that there's 6 strains of bacteria that are driving the biodynamic microbiome, and 5 that are driving the conventional microbiome. I gave you identifications of those things on the slide here. I'm presuming they don't mean anything to you. The key thing I want you to take away from this is that in all 4 of these things, sort of the 2 top hits for biodynamic and the 2 top hits for conventional, the species that we're finding are not identified, meaning that they're probably species that have never been isolated before, have never been given a name before. So your microbiomes are driven largely by species that we don't know anything about, which, right, you're chuckling. As a scientist, I think that's really, really interesting, super cool, because you probably all have bacteria and probably fungi that are new to science, that no one knows anything about them, and so we don't really know what they're doing yet. But we're starting to figure these things out, right? So now that I've done this, I know some identifications of information about these species. We can start to see whether those things are species or things that we see consistently across biodynamic vineyards. Are they things we consistently see across non-biodynamic vineyards? What kind of patterns do we get? All right, so to wrap this up, my take-home messages for you. First, microbiomes of wine grapes are highly variable, right? So what we see in one sample is going to be different than another sample. We can draw some big patterns, but everything's very variable. But sort of broadly speaking, these things are driven by geography, but farming practices plays a really big role in what kind of bacteria are present on the grapes, and therefore what bacteria go into your fermentation. Which means that the microbial terroir is something that you can, at least in part, control. So, right, lots of aspects of terroir you can't control, but this is something that you can actually have some say in, right? As you vary your farming practices, there's potential that you can influence what kind of microbes are present on those grapes, and again, if they're present on the grapes, that means they're going into your fermentation. All right, just a quick note on future research. Right, so this was, we're getting bigger and bigger, but Dr. McGuire and I just submitted another grant to the wine board, and we're hoping to collaborate in the future to do a couple things. One, we want to expand to be statewide, right? So we're not just, right, essentially, not just right around Linfield, but bigger. And we also want to start seeing whether this actually matters, right? Just because we have differences, it doesn't really matter to you unless it actually gives you results in a difference in your wine. So that's our plans for the future. And that was my thank you, and I am done. So thank you all very much.
Hi, everyone. My name is Krista McGuire, and I'm relatively new to Oregon. I've only been at U of O for a year and a half after spending almost a decade in New York City, so a little bit different. I'm going to talk to you today about one of the new research directions in our lab, looking at soil microbes in vineyards. As you all probably know, soil is the foundation of all of the vineyards, and it also contains the most diverse and abundant organisms in the ecosystem, the bacteria and the fungi. And while soil has long been known for its non-biological properties and how they relate to terroir, we're only now beginning to realize how the biological aspects of soil, the microbes in particular, are relating to terroir, as Jeremy and David have been talking about. So the research questions that we're interested in addressing in our lab is one, what are the factors that structure soil microbial communities in vineyards? I do research in a lot of different ecosystems, mostly tropical rainforests, but the overarching theme of my research is to try and understand how do plants and microbes assemble across disturbance gradients? And so agriculture obviously represents a disturbance gradient, and there are disturbance gradients within vineyards that relate to management practices. So we're really interested in finding out how does geography and climate and all those factors create communities of microbes and soils here. And then a research question we would like to tackle that we haven't started yet, but hopefully soon with collaborations with Jeremy and Greg, is how does variation in soil microbial communities ultimately influence microbial terroir? And some of the factors that we're going to be looking at as they relate to soil microbes are climate, soil type, geography, vineyard management practices, as I mentioned, vineyard varietals, so different grape varieties might be structuring the communities differently, and also seasonality. So I'm an ecologist, and so this is the ecological framework in which we are addressing and approaching these research questions from the scientific standpoint. So we're interested in three particular functional groups of microbes that you can see in the boxes on the right. We have the pathogens, which are obviously important and of interest to everyone. We have the mutualists. In particular, we're looking at mycorrhizal fungi, which form associations with most plants on the planet and almost all crops, so they're really important in the ecosystem. And decomposers, which cycle the organic material back into forms that plants can take up. And then we're interested ultimately in how the vineyard soil microbiomes respond to different factors and different management practices across seasons. Because of course, in the winter, conditions are very different than, say, in the summer, and we'd like to capture all of that variation to see what's driving the assembly patterns of these different functional groups and how that matters. So this is related to the figure that David also showed. This is from a paper in 2014 that was trying to promote this idea of microbial terroir. And this was a synthesis of all the different microbes that tend to be dominant on different parts of the grapevine and in the grapevine soil. I want to emphasize again that none of these data come from Oregon, so they're all collected from vineyards and other systems. And so we're interested in what are the vineyards like in Oregon and how are the soils assembled. And while there's been a lot of interest in how the grape microbiome is assembled, grapes are not on the vine all year round, nor are the leaves, nor are the flowers. So these parts of the plant must be recolonized by microbes from somewhere, and we think the soil is probably a likely reservoir for the microbes that are in the vineyard that then come to colonize the different parts of the plant that then may ultimately affect the fermentation and the organoleptic qualities of the wine. So to address our research questions, we set out to do a very large survey of vineyard soils across Oregon. We did a seasonal sampling throughout the year of 2018 in all 4 seasons. We sampled approximately 75 vineyards across most of the AVAs. You can see the diagram has different colored circles that correspond to different management certifications. We have the gradient from conventional all the way to biodynamic. And we use those management certifications as proxies for management because, of course, they do not always correlate. So we also sent out a survey to all the vineyard managers and asked them about particular practices, so we actually had real data and not just certification data as proxies. We sampled soils from the alley and from the vine. I'm not going to talk about those differences today, but we are finding some interesting differences between those components of the vineyard soil as well. So how do we go about analyzing soil microbes? Well, first we go out to the vineyards with our soil cores, and this research has mostly been led by my PhD student, Kay Sheck, who's in the audience today. So she can probably answer most of your questions better than I can because she's done most of the work, along with my lab technician, Tara Hebert, who's also been really helpful. They're both pictured in this picture here. And also her sister-in-law is the head winemaker at Penner-Ash, so that's been a nice connection to have. And so you'll notice in this picture that everyone's wearing gloves, and that's because we need to protect our hand bacteria from contaminating the soil samples that we take. We then take our soil sample and we place it into a sterile bag. We get sterile bags from this company called Whirlpack. And then in between each soil sample, we sterilize our soil core with ethanol. So it takes a little bit of time to sample because we have to take all these precautions to make sure we're not cross-contaminating. Then after we have our soil core, we take it back to the lab and we pass the soil core through a 2mm sterile sieve, and that's to make sure that we're getting a homogeneous sample from our core. We then extract all the DNA from our soil sample, and when we extract DNA, we get DNA from everything, right? We get the soil animals, we get the fungi, the bacteria, the algae. So then we have to selectively make millions of copies of our bacteria and fungi, which we do in the lab. And then we use supercomputer clusters to do bioinformatics and data analysis. So what have we found so far? Well, so far today, I'm going to present some of the data we have from our winter sampling effort last year. So we collected about 1,000 soil samples each season, so it's taking a long time to process those. So we only have one season of data, although our summer samples are right now in the sequencing core, so hopefully we should have those data soon. But I'm first going to talk a little bit about the data from our fungal results. So for fungi, what we can do is we can process our data through this program called FunGuild, which aligns our DNA sequences with sequences in a database and tells you whether or not they are a saprotroph, which is a decomposer, a mutualist, like a mycorrhizal fungus, or a pathogen. And so we can actually look at the relative abundance of the functional groups across the different samples that we have. So here are some of our results just to show you, give you an example of how we can do these analyses for, we have 13 of the AVAs, I believe, listed here. And you can see that the dominant functional group that we find in these systems are actually plant pathogens, which was really, at first, surprising to me. After I thought about it, it wasn't as surprising, but I'm used to working in a lot of tropical forests, and we have mostly decomposers in our samples, and mycorrhizal fungi, and a little bit of pathogens. But we have more pathogens than I have ever seen in these vineyard soils. And that makes sense from an ecological perspective because pathogens tend to be attracted to monoculture systems where you have one plant. And so that's why pathogens become so problematic, because it's a monoculture type of agriculture. So also another thing to notice is that we have a really high abundance of pathogens in areas where we have some of the highest densities of vineyards. And I think that's also interesting and important, and I'll talk a little bit about that in some future slides. So we have decomposers are the second most abundant functional group across all our samples. And then we also have a number of mycorrhizal fungi. And I assume most people are familiar with mycorrhizal fungi. I know it's part of a lot of vineyard management strategies, but for those of you who aren't familiar, mycorrhizal fungi are plant mutualists, and they help plants take up nutrients and sometimes water from the soil. So those colored things attached to the roots, that's one type of mycorrhizal fungi. And the fungi, because they're hyphae, their tissues are so small, they can extend the foraging area of the plant. The plant roots are much smaller than the hyphae. And because the fungi can't photosynthesize, they need a source of carbon. And so the plants give the fungi carbon from photosynthesis. In exchange, the fungi give the plants limiting nutrients like nitrogen and phosphorus. But these fungi are also really important in helping plants resist pathogens and other environmental stressors like drought and heat stress. So we're really interested in looking at those in our systems. And what we found so far is when we pull out the mycorrhizal fungi from our sequencing, we find that they are highly structured across the different AVAs, and they're also very highly structured across grape varieties. So in this figure here, I know that the font is probably too small to see, but essentially these are, again, NMDS plots like what Jeremy showed. Each point that you see represents all of the mycorrhizal fungi from one soil core. And the different colors represent different grape varieties next to which we sampled those soils. And the closer the points are together, the more similar those communities are. The further apart the points are, the more different those communities are. And then we use statistical techniques to assess whether those clustering patterns are significant, which they are. And so just to give you an example, you'll see in the top left the Yamhill Carlton AVA. The red points are Pinot Noir, and the blue points are mycorrhizal fungi from Chardonnay. And so the varieties, even when they are on the same rootstocks, seem to have an effect in structuring the microbial communities in the soils next to them, which was a pretty interesting result, we thought. Now, what that means in terms of function, we don't know, but that's one avenue of our future research. When we looked at soil fertility management across vineyards, we found that fertility management mattered a lot in structuring the microbial communities, but it also depended on where those communities were across a gradient of precipitation. And so what we did is we binned all of our samples into 4 different precipitation bins. We had one that was the low end, less than 40 inches per year of rain, and we used 2017 precipitation data for that. Then we had the 40 to 50 bin, and you can see that in the low bin, the fungal communities were different if people applied pruning waste versus mineral fertilizer versus compost plus pruning waste plus mineral fertilizer. And in the 40 to 50 inch precipitation bin, the communities were also structured, but they were more structured by whether or not there was pruning waste versus compost added. Now, when we get into the bin of 50 to 60 inches of precipitation, there was not much of a difference of soil fertility management on structuring the fungal communities. And this is where we have most of our samples, so we should have had the highest statistical power here. So there's clearly not an effect. However, when we go to the high end of the precipitation gradient at more than 60 inches per year, again, we find structuring of the fungal communities by soil fertility management. So there's an interaction with management and precipitation. And just to give you an example of how some of these communities are different, we have a lot of data and we don't have time to show all of it, but in the lowest bin, the less than 40 inches per year of precipitation, we pulled out all the plant pathogens, and you can see how the communities are different across the different soil fertility management types. Now, you'll see also that the general trend is the same in that the 3 pathogens that were most dominant are also the most dominant in each of these different types of soil fertility management systems. So we have Truncatella in orange, which is associated with trunk rot. We have in blue, that's Neonectria, and then the pink is Fusarium, which is associated with a lot of secondary infections, especially after phylloxera damage. Now, just because these pathogens are there doesn't mean they are pathogenic at the time. A lot of pathogens can survive as opportunists, and so they're decomposers for a little while, and then when conditions change, they become pathogens. But it was interesting that the community is structured differently based on management and precipitation, and it will be interesting to see if that does manifest into actual disease dynamics in the future. We also pulled out the decomposers, and here's an example of what that kind of analysis looks like. So you can see that the communities where we had compost plus pruning waste plus mineral fertilizer applied, we had the highest diversity of decomposers. And that makes sense in ecological theory because the more resources you give a soil community, the more niches there are for communities to thrive on. And whether or not that translates into higher function, we're beginning to explore with some other kinds of DNA technologies and substrate labeling. So soil fertility management also was very similar in how it structured bacteria communities, in that we had structuring at the low end and the high end of the continuum, but not as much in the middle 2 bins of precipitation. So you can see that here in these 2 figures, and then here's the 50 to 60 again. We have almost no structuring with soil fertility management. But then again, at more than 60 inches of precipitation, you have very strong structuring between mineral fertilizer application and compost plus pruning waste. And of course, there are different numbers and types of soil fertility management just depending on what different vineyards did in those particular precipitation bins that we were looking at. Soil tillage was another factor that we looked at, and we found that this also had different effects on soil bacteria depending on precipitation. I'm only showing you the low and the high bins right here, but you can see that the bacterial communities are quite different, whether the soil was tilled or not tilled, and tillage represents a pretty significant disturbance to microbial communities, but again, not in the middle precipitation bins. And the same was true for fungi. So again, these microbes appear to be more sensitive to soil management techniques at the extremes of the precipitation gradient. And another interesting thing we found with tillage was that in soils that were tilled, we found a higher abundance of soil pathogens. And that difference in tillage, in terms of the amount of pathogens, increased as precipitation increased. And so at the highest end of the precipitation gradient, we found a really dramatic difference in the number of pathogens between the tilled and the no-tilled soil. And that makes sense because pathogens tend to thrive in very moist conditions, but again, it seems that soil management interacts with that. And here's just, again, an example of what we find in till versus no-till. Again, different communities, as you can see, this is just at the low precipitation bin. But again, those 3 pathogens tend to be the most dominant that we're finding in the soil microbial communities. And here are the plant pathogens at the highest precipitation bin. You can see that they're quite different from the low end, right? So here's the low end. Now you move to the high end. And it seems that the Truncatella prefers the higher precipitation. And it will be interesting to, we're planning to follow up with vineyard managers to see if our abundance data for these sequence data are actually reflective of what have been historical outbreaks of disease problems on the vineyards, and then see if we can do some monitoring over time to see if they will be potential problems in the future. And I think all of this is really important to think about with respect to fluctuating climate in Oregon, especially as we heard about this morning from Greg. We are in a major drought, and it's predicted to continue to be drought conditions. And so while soil management in some areas may not seem to be a big player in terms of managing for, say, pathogens or nutrient cycling processes, if you have years where precipitation is extremely low or extremely high, then you could possibly see these differences manifest. The second research question that I said we're interested in addressing is how the soil microbiome relates to microbial terroir. And we do think that the soil microbiome is probably a big reservoir for a lot of these microbes that end up in the fermentation vessels. And so what we did is we pulled out some of the microbes we know are involved in adding organoleptic properties to wine, just from the literature from previous studies. And we just looked to see if they were in our soil data. This is not a comprehensive survey yet. We're going to keep adding and doing more searches, but we found quite an abundance of a number of them, that the bacteria are on the right and the fungi are on the left. So we have some bacteria that are involved in malolactic fermentation, and then some of these other yeasts that are not Saccharomyces cerevisiae. And it'll be interesting to compare our data set with Jeremy's data set, which we're planning to do because a few of our sites actually overlap, where we can look at the grape microbiome and the soil microbiome together to see how similar the communities are, and if we can detect whether or not the inoculum on the soil or on the grapevine grape clusters is actually coming from the soil. Similarly, we pulled out some of the microbes that we know are involved in wine spoilage, and we did also find some of those in our microbial sequence data from the soil. So we're going to continue to look for more of those and add to our database, and also see how these change across the different seasons. So one aspect that we're also interested in considering is how these microbes are moving around vineyards. So in ecology, in ecological theory, dispersal is a really important aspect of how microbes assemble. So if you have a vineyard in the middle of, say, a fragmented landscape, the vineyard size, its degree of isolation from other similar habitats, and the quality of the surrounding habitat are really important and can really be predictors of how microbes may or may not be able to disperse and assemble in the particular vineyard. However, if you have a habitat where you have a really high density of vineyards, things may be able to move around a lot more. This is a theory called meta-community dynamics in ecology, and it's something that we are starting to explore in this system. And we actually did some preliminary analyses and developed a device where we can now sample microbes from the air as they're moving around. And so we're going to do, hopefully, an experiment where we can look at how the microbes are moving around from vineyard to vineyard, and how that's dependent on all these different factors that we've been talking about. And so that's my student, Kayla Evans, who was setting up this project in a couple of vineyards by us. And finally, we're hoping that eventually we can get enough funding so that we can put all of this together, and we can look at how do the different aspects of climate and geography and management influence the assembly and the function of the soil microbiome. How does that influence the assembly of the microbes on different parts of the grapevine? And then ultimately, how does that influence the flavor profiles of the wine? So just to summarize, we've found that regional variation in soil and climate result in different assemblages of soil, fungi, and bacteria, which may affect microbial terroir. So we did find that in the different AVAs, we had very different assemblages of fungi and bacteria for all the different AVAs so far that we've sampled. Management practices locally seem to further influence the local communities of fungi and bacteria, which may further influence microbial terroir. And mean annual precipitation values that we've looked at seem to interact significantly with management practices, which has implications for climate change effects and climate fluctuations. And with that, I would like to thank all of the vineyard managers and winemakers and vineyard owners we've worked with, Everyone has been really helpful and collaborative and very generous in giving us access to sites and information about these systems and helping us design our sampling strategies. And I'd also like to thank the Oregon Wine Board for inviting me here to talk today. Thank you.
Good stuff all the way around. My job here is really in about the next 5 minutes to put a global perspective on this. And I'm coming at this from a couple of different angles. I've been involved with terroir-based research for a number of years. I'm on the board of directors of a couple of international congresses that we are seeing increasing interest in this area. I'm also functioning at different levels within international journals, and I want to talk a little bit about kind of what's happening within that framework of looking at microbial research within the terroir frame. So, some of this is a little redundant, of course, with what Jeremy and Krista presented, but, this is pretty straightforward. All plants and all environments depend upon microbes. We need to better understand it. I think that all crops would benefit clearly, but in this industry, we already have a keen interest in it, so we need to be able to understand how to categorize our microbial environment, but also optimize it in ways that can be beneficial to our product. This top title comes from my friend Jamie Good. I heard him say this at a conference not long ago. No microbes, no wine, period. So wouldn't that make sense that we understand this better? A real complex set of questions can come from this because this community is complex, but it's at the heart of what we do in producing wine. Nutrient issues within the soil, clearly, plant health through protection against pathogens, as Krista mentioned. Also mitigating environmental stresses. There's some interesting research going on in a few different places that have looked at this already and having some very interesting results. But also understanding fermentation, of course, through the action of yeast within the microbial community. So a lot of interest is going on out there in the world from wine growers, technical institutes, the scientific community to better understand this ecosystem overall. And I just wanted to put some perspective on this, talking with my colleagues and being out there in the world of this. First of all, the OIV, which is the governing body for viticulture and wine production worldwide. The U.S. is not a member of it, but that doesn't really matter because the OIV is seeing this as a very important topic, one that they're considering adding as a priority theme in their research agendas for both viticulture and wine production. Wine Australia has increasingly funded microbiome research, and they're planning on investing more in it. The AWRI is conducting extensive bacteria and yeast studies currently. INRA in France is facilitating numerous regional studies, and it very much has a terroir-based focus in France, you can imagine. Microwine, if you haven't looked at this, really a good group. They have a strong web presence. It's a collaboration between industry and academic partners to examine the role of the microbial community in wine production, but it's also a clearinghouse for a lot of really great information on this topic. The Institute of Vine and Wine Sciences in Spain has developed a research group looking at this as well. The Viticulture and Enology Research Center in Italy is also doing some work here, looking at this across their different terroirs in the country. The Catina Institute in Argentina has funded a few projects that have been very fruitful overall. They've even gone so far in the Catina Wine Company to name one of their wines after one of their dominant microbial units. The New Zealand wine growers and plant food research have also funded significant research. There's 2 PhD projects and a master's project going on right now looking at microbial components of terroir in New Zealand. Microbiome research has also become important in numerous terroir-based studies that are out there. Large wine companies such as Sograp and Torres have R&D programs that aim to use this to better manage soil health and biodiversity, promoting plant resiliency. International journals in viticulture and enology that I've been a part of are considering microbiome research as hot topical areas because they're seeing more and more submissions and publications within those areas. The journal Frontiers in Microbiology is also considering submissions from viticulture and enology groups as hot areas within their journal in terms of manuscripts. And major international conferences, at least the two that I'm involved with right now, the Terroir Congress and JESCO, are both focusing at least one session at each one of their meetings on microbial terroir aspects. So I think, you know, from what we saw here in Oregon, this is a great start, but all of this is really still in its infancy. It's growing rapidly, I think, throughout the world. I think we're going to see more and more interest. As we learn more, I think that will pique even more interest down the road. Better and more sophisticated methods of analysis are coming along as well, and as we develop those, I think that more interesting and promising results will come from it. I think the key really does appear to develop a further understanding of what the functional meaning of the microbiome means in terroir expression. If we can come up with ways in which we can define that functional meaning of that, I think we're going to really succeed here. Overall, I think the goal should be to develop a better understanding of the microbiome such that it allows all of us to grow better grapes, make better wine, and be better stewards of our environment, and ultimately more sustainable in the long run. So with that, thank you very much.
Thank you, Jeremy, Krista, and Greg. You've heard about both soils and what's on the grapes. You've heard about the AVA, the geographic distribution versus the distribution in other columns, such as with respect to water availability and so forth. You've heard about some of the interest in microbiome work that's going on around the world. And so the fact that we're really now beginning to explore the microbiome in Oregon is, I think, a key step to becoming a part of this larger international community. And we now have some time for some questions. I think there is something here on the iPad, and if you want to ask questions, there are also, we can't see them up here because the lights are very bright, but there are microphones on stands there if you want to ask a question. There's a question here while we're waiting to see if anyone wants to follow up on a live presence. Did any of your study sites return pumice to their vineyard, compost it or not, and do you think this practice would affect the microbiome? Jeremy, Krista?
Kay is shaking her head yes. So I would direct that question to Kay because she is.
Is that micro?
Oh, sorry, this is on. Yes, Kay is shaking her head yes. She has all the survey data on the particular practices of the vineyards. How many of those vineyards would you say, Kay? 10, maybe? Oh, half of the sites? Okay.
And
so
was
yes.
was that composted?
And yeah, it was composted. Yep.
Jeremy, do you know if your sites?
I honestly have no idea.
Okay.
I don't.
Okay.
We didn't survey as extensively as.
Yes. Okay. Is there a question right there?
Yes, I have a question. Hi, my name is Rebecca Sweet. I have a lot of questions, but the one I want to ask today is, I think that the focus on this research on terroir is really interesting and important, but as a grower, and speaking on behalf of the growers, I think a more interesting question is how does this influence our disease management practices? This is something that we think about daily, and a lot of our inputs and time and money goes towards this as growers. So that's my question.
Who would like to address that?
Sure, I can speak to that. Yeah, this is a question that we're really interested in addressing as well because we were talking a lot with the vineyard managers and they had expressed a very strong interest in trying to understand this, and that's why we're looking specifically at pathogens as it relates to the different practices across the AVAs. And we're hoping to set up an experiment where we're actually going to be manipulating the mycorrhizal fungi of the vine and then giving pathogen challenges to the plant to see if that would actually help in disease management. So that's one aspect of disease management, but that's something where we're interested in because a lot of these mycorrhizal fungi can actually induce systemic resistance in the plant that we can measure. And this may be context dependent, right? It could be dependent on the soil. It could be dependent on the climate. And so we're hoping to tease apart some of those variables. The problem is we don't have baseline survey data in Oregon yet. To get there, we want to get there, and I think all of us are really interested in getting there in addition to looking at how it influences terroir. But right now we're trying to get a sense for what is the landscape, how are the microbes structured, can we use statistical techniques to partition out the different variables that are influencing how the pathogens are structured, and then can we actually do experiments to look at the mechanisms. And so that is definitely on the horizon and something we're interested in. It's just going to take a lot of groundwork to get there, first to know who is there and how are they assembled. But yeah, thanks for that comment. It's really important.
Jeremy, do you want to add anything to that? Okay. I see a question right here.
Yeah, I have a comment and a question. Both the researchers divided farming practices into, conventional, live, organic, biodynamic. And there's a hybrid out there which is live, that does not use herbicides and minimizes sulfur use. And my specific question is, has any research that you've done or that you've seen show the impact of sulfur use on the microbiome? Because a lot of times when you go to an organic or a biodynamic vineyard, you end up having a higher usage of sulfur to deal with the fungus in those vineyards.
So short answer, no, I don't think anyone's done that yet, but that's a really good point and a really good question. It's certainly sort of why I sort of started asking things the way I did, right? I mean, I said I did 40 vineyards and then I did 10 vineyards, and we want to just keep expanding that sampling and start trying to look at, to really drill down on what does farming practice really mean and what impacts does that actually have. So yeah, I hope that we'll get there in the future, but we're not there yet.
Yeah, and for our samples, that's one of the factors that we are analyzing our data against. We don't have those analyses done yet, but that's something that we are looking at. I know that in our preliminary analyses, we also did this technique. It's called shotgun DNA metagenomics. It's kind of a mouthful, but essentially we take the DNA of our sample and you essentially blow it up into little bits, and then supercomputers put all the clusters together, and you can look at all of the different organisms in your sample, all of the different pathways. And we found some correlations with sulfur metabolism genes and biodynamic vineyards, because I think there is probably more sulfur used there. And so that's one of the things that we decided we wanted to look into a little bit further, because there were a lot of genes that were involved in metabolizing sulfur, which probably means there are different kinds of organisms that are metabolizing sulfur in those soils. So that's something that we will be looking at in the future.
Just to add one more thing, I want to be really clear here that right, everybody, you all have your sort of preferred farming techniques, and we certainly don't know yet. I don't think I would ever want to say that what you're doing is good or bad for the microbiome, right? I mean, you're doing things to get the wine that you want. So I hope to sort of tell you whether what you're doing has an impact or doesn't have an impact, both on sort of all of these factors, but I am very careful to not place judgment on this, and I don't think any of you should either, right? A microbiome is what it is, and it's a really, really complex thing, and so again, it's not like good, bad, whatever, it's just, it's there, and I hope that I can help you figure out what you have, and then you can decide whether you're happy with that, whether you like it, what do you want to do with that information.
I think that's an extremely important point, and one of the things that fascinates me about this study is the notion that you can compare farming practices, in particular the biodynamic technique of spraying teas. And there's a question here from someone who asked whether you've looked at some of the, at least in the case of Jeremy, with regard to what you find on the grapes, although maybe also in the soil too, Krista, whether any of those bear any particular relationship to what is typically, I know teas vary and depend on, timing and spraying and so forth. How does that compare to what might be, shall we say, inoculated from the teas?
We have some samples of teas. I see Doug Tennell sitting over there. He's happily shared some teas with us, and we have those samples, but I don't have that data analyzed yet, so I don't know. But it's definitely something that we've thought about. Like, do I know, is this thing that you're putting out what we then find on the grapes? But
Yeah, we have actually in our sequencing run right now, we have some compost samples that we're sequencing. We don't have tea samples, but that's something that we have thought about, and I think in our future runs, we would like to do that. But we were sampling so many vineyards that we couldn't go deep into any vineyard. We were going very broad. So in the future, when we go deep into particular vineyards, that's something that we would definitely incorporate into our analyses.
So one of the menaces that we all face in the vineyard are trunk diseases, and I think there's a whole host of various fungi and bacteria both that cause these. Do you think that to what extent are those contributed by the soil? And do you see any relationship? I know in this case, Krista, since you're looking at the soil, what do you see a relationship between the organisms found in trunk diseases? Or maybe this is a premature question and something that you would like to explore, but do you think there's a relationship between what you see in the soil with what we find causing disease in the trunk, in pathological conditions in the wood itself?
Yeah, so in our data set that I showed with the pathogen stacked bar graphs, we found Truncatello, which is one of the ones associated with trunk disease, was our most abundant pathogen across all of our samples in all AVAs and all management practices. It varied in how abundant it was depending on the precipitation and to some extent the soil management practice that we were looking at, but it was overall the most abundant. So it doesn't surprise me that it's one of the most important diseases, and it was interesting that our sequence data was actually reflecting that disease, which is pretty prevalent here. So it'll be interesting to see if these abundances where we're finding Truncatello and some of the other organisms associated with the trunk disease, where they're even higher relative to other vineyards, if it becomes a more problematic disease versus some of the other vineyards where we have them as lower abundance. Of course, that could also depend on some of the other microbes that interact with the pathogens. But.
I see some folks over here.
Dr. Weitz, do you know if the biodynamic vineyards in your study sprayed with a compost tea or other bacterial sprays, and if any of the conventional vineyards sprayed with that, and could that be a factor in the diversity? It might be interesting to do a conventional vineyard with a spray like that to see whether it's that they're getting inoculated or whether it's the synthetics that are sprayed that are kind of bringing it down.
yeah, I don't definitively know. I believe that all of the biodynamics that we sampled are using teas, and I believe that all of the non-biodynamics are not using teas. I'm not 100% positive on that, but I think so. But I would also, right, we tried to pair things that were as close to each other as they can, so there's also some possibility of drift, right, as Krista talked about, right? Mm-hmm. These microbes are definitely going to disperse, so what's happening, say, on one side of the hill might be drifting over to the other side of the hill and could certainly impact those microbiomes, so.
Kevin?
Is it not the case that when we're doing DNA analysis, we might actually be looking at a lot of organisms that have been dead, some for extended periods of time, and have no impact whatsoever in the environment at that time? And to that, I suggest, should we not perhaps be considering RNA analysis instead?
Yeah, there is this thing called relic DNA, and I know what you're talking about, where this one study came out finding that a lot of DNA, well, I guess it was about 10 to 40% of the DNA in some of these DNA studies was technically dead, and that it could persist in the environment for quite a bit of time. That's a bit controversial right now in the microbial community, and I think it also depends on the ecosystem, how long that DNA can hang around. I am a skeptic of how long that DNA can hang around in disturbed ecosystems, like an agricultural site. I also think that even if you're finding DNA that is not living at the time, it means that that organism was reproducing at some point and it died, and a lot of these microbes have really high turnover times. And so even if you're finding DNA that's not living, it probably means that there's the potential for that lineage to be alive and functional at some point in time. But that caveat said, yes, RNA is a much more direct way to go about looking at the function, and that's a direction that we're going in some of Kay's work that she's going to be doing this summer and in coming years is going to be doing RNA transcriptomics is what we call it, to actually get at what microbes are functioning and how does that depend on particular contexts or particular substrates that we're adding to them or particular mycorrhizal fungi that they have. So yeah, so that's a great point, and that's something that we are going to look at. But I don't think the DNA data are completely crap data. I think they do still have a lot of relevance, but I think that RNA data are important to couple with the DNA data to look at the full community. Yeah.
We have time for one last question. There are lots more on this iPad. I'm sorry we can't get to all of them, but for Krista, since you were interested in what grew in the various water column water availability systems, did you look at compare any of it to specifically irrigated vineyards?
No, and that's something we are going to look at because some of the vineyards we know, and especially the really dry climates, do have some drip irrigation. So we are going to look at that. We just haven't had a chance to look at those data yet. But yeah, that's an interesting point.
This is obviously a topic that we will hear a lot more about. Based on what Greg said, we'll hear a lot about it from around the world. Thank you all for being here to help us start this exploration, specifically with regard to Oregon. Thanks for coming.